We continue our journey about the origin of life. Just to quickly recap, we stated that we began at the chemical evolution state, in which we had simple chemical molecules that eventually turned into smaller organic molecules, and then we just saw how those small organic molecules eventually formed into larger organic macromolecules. The next stepwise process in this origin of life is now the formation of what are protocells. So we'll write, this is going to be entitled our protocell formation. And we talked about protocells very briefly in our chemical evolution flowchart. Protocells, proto, is the Greek term for first cell formation. Before we get into the actual formation of these protocells, one thing we have to make sure we understand is the concept of a vesicle. What is a vesicle and how does it relate to the life and the origin of life? A vesicle is defined as something, think of it as a fluid-filled compartment. Simple as that. It's a fluid-filled compartment, and more specifically, it's actually surrounded by a membrane. It's also surrounded by, let's say, a membrane, M-E-M. What a good example of a vesicle you can think of is, let's say, imagine a drop of oil in water. That'll give you a good idea of a vesicle. That oil stays completely separate from the water because it surrounds itself. It has this hydrophobic layer that's surrounding itself like a membrane. And inside of that hydrophobic layer are those oil molecules that are keeping itself a distance away from the water molecules because of that hydrophilic nature. So oil and water is a good way to think of vesicles. So keep that in mind as we continue our discussion. So let's actually talk about protocells in a little more detail. So let's look at protocell. What is a protocell? We talked about this originally. A protocell is just very simply defined as an aggregate. Aggregate just means a combination of abiotically. What does abiotic mean again? Without, and bio refers to life. So non-living, aggregate of non-living produced organic polymers. So what happened in our stepwise sort of journey of origin of life? We started with the chemicals, then we ended up with small organic molecules, then we ended up with larger organic polymers. Look what we're doing now. We're combining all those organic polymers that originally came from abiotic origins and aggregating them, putting them together into a protocell. So one thing we just want to make sure we understand is that a protocell is not produced by a living organism. It has abiotic origins that are not from life, not produced by living organism. So the first cell was not produced by a living organism because if we go back, we had to evolve chemically, then we had to create a small organic molecule, then we had to create a larger macro-organic molecule, and now we're combining those polymers into one protocell. Once again, this is all abiotic. Now, a protocell, interestingly enough, is the first point at which we start to exhibit life's characteristics. So it exhibits living cell characteristics. What do I mean by this? Certain characteristics of protocells that are observed show that it is life, that it is the sort of starting point and the true origin of life. Things like an electrical potential are seen in protocells. Simply put, an electrical potential just means that the inside has a different, let's say, electricity level than the outside. There's some sort of gradient involved. We don't need to get into those details. Just know that there's an electrical potential, and that's something seen in life, especially living cells. In addition, we notice that protocells do something very interesting. They actually, what is known as, absorbs materials osmotically. Think of it like this. What these guys do, these protocells, they see water. They have the ability to either shrink and give water, let's say, away or out into their environment, or if they see water and they're, let's say, need it, they can swell. So they're shrinking and swelling. It has the ability to act with osmosis, to transfer water. That's what osmosis is, the transfer of water either outside or inside the, let's say, enclosed vesicle. So that's another life characteristic we start to observe. The most important one, I think, is this idea of an internal environment. We touched upon this in our earlier videos. The internal environment in a protocell is not equal to its external environment. What does that mean? Basically, the inside is independent of the outside. The inside can finally, we haven't seen this before, act independently of what's going on the outside. Let me just rewrite this a little clearer. External, ENV, environment. So the outside is completely independent of the inside and vice versa. This is one of the most important ones that's often seen on exams. This is the first sort of life characteristic that's very important in protocells. In addition, what we notice is that when the protocell is increasing in size, it actually divides in half. Divides in half when big. So this is an interesting concept, that when it gets bigger, it actually starts to replicate itself. But one thing you want to make sure about this is that we still don't see heredity. So make sure you understand this. There is still no heredity. Heredity is defined as the transmission of genetic information. So we can write that down. It can't transmit, it meaning the protocell, genetic info to next gen. So when it splits, it does not transmit any sort of information telling the next generation this is how you're going to evolve and grow. So this is our introduction to what a protocell is and how it forms. In our next video, we're going to be looking more specifically at this idea of heredity and genetic material and seeing how the protocell eventually gets this ability to do this, to use genetic information and pass it on to the next generation.